Well, hello there. Do you ever wonder how organisms grow? We start as babies that crawl and yell, then to children that run and yell, then up to adults, which have responsibilities. But how do we grow? How do we gain height? How do we gain weight? When you have injuries or cuts, how do those wounds heal? How does an entire person come to be from a single cell after fertilization? What do you think so? Now, we're going to answer those questions, and those questions are answered by the process of mitosis. So what is mitosis? A brief history lesson first. So the separation of cells to form daughter cells were first described by Otto Bütschli. This was back in 1870s. Another scientist is you have this guy. This is Walter Fleming, where in 1879 he first coined the term mitosis, which came from the Greek word mitos, meaning threads. This is to describe the appearance of chromosomes under the microscope. Mitosis is the biological process whereby a cell divides into two daughter cells. So this is done by replicating its genetic material along with the separation process that produces two cells identical to each other and its parent, essentially being clones of each other. You have to remember that mitosis is only a small portion of the entire cell cycle, and its importance lies in the final event on cell division. The first phase of mitosis is prophase. Remember the letter P, and it stands for preparation. So this is the initial stage wherein chromatin condenses to form visible chromosomes. So these chromosomes were replicated and appear as two sister chromatids joined by the centromere complex. The next stage we have metaphase. Remember the letter M stands for middle. So during the metaphase, the chromosomes line up in the cellular equator on the metaphase plate, and microtubules from the poles attach to the kinetochores of each chromosome. On to the next stage, you have anaphase. So for anaphase, the letter A, remember A for apart or away. So during the anaphase, the microtubules contract and break apart sister chromatids by pulling the kinetochores towards the cellular poles. The last stage before total separation is we have telophase. Remember the letter T stands for either twins or the number 2. So during telophase, the separated chromosomes on both poles decondense and the nuclear envelope starts to form and begins the separation step via the next stage, which is cytokinesis. So cytokinesis is the culminating stage in cell division. And cytokinesis is where the cytoplasm is divided into two during the formation of the cleavage furrow. This cleavage will then form and break to result two identical daughter cells, clones of each other and that of the parent. The visualization of mitosis can be done by conducting the famous onion root tip experiment, where sections of the onion root tip are cut, stained, and viewed on a microscope and easily observe different phases of mitosis on the plant cell. To test your knowledge, what do you think is the phase of mitosis this cell is undertaking? If you said prophase, you're right. Next, what about this cell? If you said metaphase, that's correct. How about this one? This next one? I'll give you a clue. They are being separated apart. What do you think? If you said anaphase, then you're right again. Then the last would be this one. What do you think this phase is? If you said telophase, then you're correct. Overall, this basic and fundamental biological process determines the growth and continuation of life by creating more of life's building blocks, which are cells. I hope you enjoyed this short video explaining mitosis and you learned something from today. Thanks for watching!